Hi students, welcome back to Gene Learning. Yashwant here again. So in the last class, we were talking about sexual reproduction in flowering plants and we had just gone through the introduction. So in this class also, let me just take a few minutes to recollect what we had just, done in the last class. In the last class, we were talking about the different types of reproduction which takes place and what are the types of gametes which are involved, what are gonads, etc. Now, also we discussed that in the chapter Sexual Reproduction in Flowering Plants, we see that there are five subtopics which we have to discuss. So it starts from the flower. Now, flower is the reproductive structure of a plant. Now, this structure is created with a lot of processes involved that we'll discuss a little later. So the structure is created for the purpose of reproduction and to continue their progeny. Okay. Now, these are the fascinating organs of the angiosperms or the flowering plants, right? Next, it's pre-fertilization structure and events. Now, in this subtopic, what we see is that what are all the structures which are involved and what are the events which takes place for the formation of the inflorescence or the flower. Next, we'll see that what are the things that are formed from the structure and events. Okay, what has happened, and from there the fertilization which takes place after the pollination. And what is double fertilization? Now double fertilization, as far as my knowledge, it's the best, or what I can say is a unique, beautiful process which takes place only in angiosperms. Okay, so that we shall discuss in this double fertilization. Next, after that, what follows on is that post fertilization structures and events. So in this, we'll see a lot of differences right from the structures from pre fertilization and the changes which takes place in the double fertilization. And after that, in the post fertilization, what we see is totally different. So that different structures and events. So the formation of structures and what are the events which led to the formation of structures, we see that in the post-fertilization subtopic. Then finally, we talk about apomixis and polyembryony. As we all know that apomixis is a type of asexual reproduction which mimics sexual reproduction. Okay, and polyembryony is the formation of many embryos in a single ovary. Okay, so that we shall discuss in this chapter. Okay, now basically, what do you mean by a flower? As we all know, that flower is a beautiful structure, okay, which has been created by a plant. So, in that sense, what does it mean? Does it mean that flowers are only developed for the extra ambience created for human beings to enjoy, and they give out very good fragrance, and we use them in different occasions? No, angiosperms or the flowering plants produce flowers for the sole purpose of reproduction. Okay. Now we see that there are different structures are involved in this formation of this flower. Okay. So as I told you, inflorescence itself is a type of a process wherein we see that the apical region of the plant or a tree gets converted into an inflorescence due to the activity of different hormones in the plant body. Now, we see that flowers are, what can I say is that we see flowers throughout the year, right? And also, we see some flowers only at certain time of the year. Now, what is the difference? The difference is such that the flowers are there, either the plant continues its reproduction till the end, okay, of its living, or some prefer to only flower only when they feel that the atmospheric conditions, okay, in which niche they are there, so that provides them an opportunity for developing inflorescence and from there carrying out sexual reproduction. Okay, so that is what flower is for in a plant. Okay. Now, any plant, if you take that plant, if it gives out a flower, then that plant is definitely uh, uses sexual reproduction as one of the means of reproduction, or to develop fruit, or to develop seed, and both are one and the same. See, for a plant, if it develops a fruit, fruit is not of importance to it. Only the seed is. Fruit is a bait. Okay. So that all we'll discuss little later. But flower is there for that reason. Okay. Now let us talk about different organs. Okay, in the flower, which leads to the act called as the sexual reproduction. Right. Okay, that we'll see now. As I was talking to you about the structure of a flower. Now, before that, let us see that, you know, uh, what are all the flowers? The flowers are nothing but, okay. Now, we know that human beings have a very special relationship with the flower. Now, what do I mean by that? Very simple. Any occasion, if you take, human beings, as we use flowers to enrich the value of that occasion. For example, marriage. So in India, especially, no marriage is conducted without flowers. Okay, it might be a housewarming ceremony, it might be someone's birthday, and even if someone dies, we need flower. But as we all know that flower is not generated by or produced by a plant for our, you know, personal needs. Okay, flower is produced as a sexual reproductive, what do you say, organ. Okay. See, as we were talking about different types of flowers and fragrance and everything, we also see some kind of a difference in some of the angiosperms, and it is quite peculiar to see that also. Now, what do I mean by that? When we look at a flower, okay, now pick a flower for that matter. Now, if I take a hibiscus and if I look into it, I see that both male and female reproductive structures are present in the same flower. Whereas if I see another plant, for example, where I see that the flowers which are produced in that plant are either male or female. Now, what do I mean by this? I mean to say that in the same plant, we find a male flower separate and a female flower separate. And there is pollination between them. All that is said and done. And there is another type which is even more peculiar. Now we call those plants either male plants or female plants. Now what is this all about? Yes, the reason why they call that a male flower, male plant or female plant rather, is because of the flowers which are produced in that particular plant. Now in a particular plant, if we find only female flowers, then that is a female plant. And if we find a plant with only male flowers, okay, so those are called as the male plants. Example is your papaya. Papaya is a very good example for this. So we find a male papaya plant as well as a female papaya plant. Okay. Now, without any further delay, let us talk about the different parts of a reproductive, parts and accessory parts of a flower. Okay. Now here we have a diagram where it says that it's the LS of a flower. Now, if you take a typical bisexual flower like this, we find different organs. Like I've wrote few symbols over here, two symbols. One is this is a female symbol, and this is a male female symbol. Okay. Now, the reason why I've written it is that because of the different parts which we find here. We find, in as I told you earlier, both the part, that is the male reproductive parts or structures, and there also we find female reproductive structures. Now let's talk about it. Now, when you look at this, okay, reproductive structures are different, and accessory parts of the flowers are also different. Now, when we have to look at the accessory structures in a flower, is that we find the sepals and as well as the petals. Now, usually when you look at the sepal of a flower, it will be greenish itself, and petals will be of varied colors, very attractive, etc., etc. But the important structures in a flower are the male and female reproductive structures. Now, when I tell you about a female uh, structure of a flower, means it is called as nothing but pistil, and these are the structures. Okay, and yeah, right. So, what are the different parts of this pistil? Now, this is the pistil. Now, pistil consists of three parts, that is, stigma, style, and ovary. Now, what is stigma? Stigma is a receptacle where the pollen grains come and stick, and that too, as these flowers are exposed to the air, any type of pollen grain might come and stick to it. But there are, chemical or chemotactic reactions which takes place between the chemicals or the nectar or whatever is present in the stigma on the stigma with the pollen grains. Okay, I'll give you a scenario. Now, imagine a pollen grain from the same plant come and stick to the stigma. Okay. Now, that pollen grain has the chemo thing or the chemotactic reaction thing happening fine. And from that, there is a pollen tube which is running down, or it will be developed from the pollen grain, and it runs down through the style and it reaches the ovary. Okay. Now, in the ovary, we find ovule, and the ovule guides the pollen tube inside it so that the gametes which are present, there will be two gametes. I'll tell you about it in a moment when I talk to you about the stamen. These gametes are guided inside this ovule, and their and the fertilization action takes place. Okay, so in the stigma we find that kind of a reaction taking place, and the style is the region where the pollen tube gets developed and it reaches the ovary. So ovary is a place where you find the ovule. Okay, so that is the different parts of a pistil. Now, when you talk about the male region or the male structure of a flower, it is nothing but stamen. Okay. Now, the stamen consists of two parts, right? So, it consists of anther, and it consists of a stalk-like structure called, excuse me, stamen consists of anther and filament. Now, anther is the place where these pollen grains are developed. Further details into it we'll study a little later. Okay, and the stalk like structure which is holding it is called as the filament. So, altogether, this is the male part which is called as the stamen, male structure in the flower. Okay, now this, as I told you earlier, is the place where inside which the pollen grains are developed, and there is a special action which takes place over there. That means when the pollen grains have matured inside this anther, there will be something called as the dehiscence which happens, like either a vertical slit, or at the top there will be an opening. This you have studied in your first year PUC. So dehiscence is a process where the cells lose water, and there is this opening which is created from the anther so that the mature pollen grains start to go out, or it might stick to an insect or a bird or something, but it reaches the stigma. Okay, so the pollen grains purpose is that. So pollen grains, as we all know, it has two regions, intine and extine. Yeah, that we shall discuss little later. Okay, so that is again from your first year PUC. Right. Now, as we know that now, these are the different parts of the typical angiosperm flower. Now, this is the LS of a flower, that is the lateral section where we study about the different structures. This is a bisexual flower. When I say bisexual flower, it means that both the male and female reproductive structures are present in the same flower itself. Okay, so this we find stamen as well as pistil, collectively called. Okay, so this is about the flower. Okay.
Now let us talk about the second subtopic, that is prefertilization, structure and events. Before a plant could develop a flower, there would be a lot of internal changes with respect to hormones and other reactions, which are the real cause for the production of flowers. Now, typically, what happens here is that the apical region of a plant, you see that the formation of inflorescence. Now, this inflorescence initially consists of floral buds, and inside that floral bud, we find the formation of male and female reproductive structures. So, male reproductive structure here is called as the andrecium, and female reproductive structure is called as gynecium. So basically here andrecium is nothing but a stamen and gynecium is nothing but a pistil. Okay. Now, when we have to talk about the andrecium, first we'll talk about the andrecium, then go to the gynecium. Here we see two different diagrams where in this diagram we see a typical stamen, and here we see the 3D structure of a as I can say, I cannot say that it's a transverse section altogether, but a three-dimensional structure depicting the inner parts of the anther. Okay. Now look at the stamen. Stamen consists of two different parts. As we have discussed this already, but let us go through it again. It consists of an anterior anther and a stalk-like filament. Now, when we talk about anther, okay, a typical anther is a bilobe structure. Now, what do I mean by a bilobe structure? Here you see this lobe, this is one lobe, and the other one is adjacent to it. Now, both are attached to each other. Now, this is one lobe and this is another lobe. Altogether, we call it as a bilobe structure. Now, this bilobe structure, that is anther, is made up of two theca. That is what we call it as dithecous. Dithecous. Okay. So this, if we had to take a three-dimensional structure and see that, we find different regions. I'll come to it in a short while. Let us talk about the filament. Filament is a stalk, like as I told you earlier, a stalk-like structure which is attached to the base of an anther, and the base of the filament is attached to either the thalamus or the petal of a typical flower. Okay. Now, if we had to take different types of stamens and compare it, the size, shape, and the colors would be very different. Now, if you are an inquisitive student, I say. Now, what I ask you to do is kindly go search around for 5 or 6 different types of flowers. Kindly pick a single stamen from each of it, arrange them on a piece of paper, and you can make out that difference very easily, that some of the stamens are very small and some of them are very huge in the terms of anther as well as the filament. Okay, and even the color, it varies. Okay, now when you look at anther, okay, anther, as we have already seen, that it's a bilobed structure. But if we had to take a transverse section of this anther and look at it, now it reveals a different structure altogether. Now, as you can see here, it looks like a four-chambered structure or a tetragonal structure. Now, what are these four chambers called as? They are called as microsporangia. They're called as microsporangia, and this microsporangia later develops into what is known as the pollen sacs. Now, as we can already see from the diagram, that this tetragonal structure, that is the microsporangia, there are four microsporangia, each one in the corner. Okay, tetragonal structure, as I've already told you. So this becomes what is known as the pollen sac little later, pollen sacs, and this pollen sacs, all through the length of these lobed structures, that, as I told you, bilobed structure, it consists of millions and millions of pollen grains. Okay, once when the pollen grains mature, what happens is that, as you can see over here, that there is something known as the line of dehiscence in each of the lobe. In each of the lobe, we find what is known as the line of dehiscence, and this line of dehiscence opens up for the mature pollen grains to go out or get dispersed, so that it gets stuck to the stamen for further reproductive activity. Now, let us talk about structure of microsporangium inside an anther. Now, for that, first let us take the TS of an anther, that is the transverse section of an anther, young anther. We will take the TS of a young anther, where we see that there are four microsporangium, as we have already discussed, in the corners of the anther. They are situated over there. Okay. Now this microsporangium is surrounded by four layers, that is outer epidermis, inner endothecium, and before that we have middle layers, and the fourth layer before middle layer is called as the tapetum. Now tapetum is a layer which consists of a lot of nourishment stuff. Now this tapetum is known to nourish the developing pollen grains. Okay, now that is the function of the tapetum. Now the function of these three layers which are surrounding the microsporangium, as we can see over here, is that they help in protecting the microsporangium, and also they help in during the time of dehiscence. Okay, now let us look at the enhanced view of the microsporangium. Here also we find that the first layer would be epidermis, then endothecium, middle layers, then you have the microspore mother cells over here, then you have the tapetum. As I told you, tapetum is a very dense layer. The function of the tapetum is to nourish the developing pollen grains. Okay, now if you look at it carefully, that is the TS of young anther, you find in the middle of the microsporangia you find dense cells. Now these dense cells are called as the sporogenous tissues. Okay, now these sporogenous tissue, each cell of the sporogenous tissue has a potential to become a pollen grain later. Okay. Now, if we take this sporogenous tissue and look at it carefully, each cell of the sporogenous tissue is called as microspore, spore, or pollen mother cell. Okay. Now each cell in this sporogenous tissue is called as microspore mother cell or pollen mother cell. Now this microspore mother cell or pollen mother cell, if it divides meiotically, they have the potential to give rise to what is known as the microspore tetrads. Microspore tetrads. Okay. Now, this is nothing but microsporogenesis. What I just said is nothing but microsporogenesis. That is, if you want to define microsporogenesis, it is nothing but the formation of microspore tetrad from microspore mother cell through the process of meiosis is called microsporogenesis. I'll repeat again, microsporogenesis is a process where each microspore mother cell from the sporogenous tissue, okay, divides meiotically to form microspore tetrad. Okay, so this is called as microsporogenesis. Now, as and when the anther starts to mature, okay, and it starts to lose water. Now, as this process starts to happen, what happens is that this microspore tetrad starts to dissociate. Now, what do you mean by this microspore tetrad? It is nothing but each microspore mother cell, when it divides meiotically, it gives out four microspores which are clubbed together. That is what we call it as microspore tetrad. So this is microspore tetrad. Okay, so from the single cell of a sporogenous tissue, which is called as the microspore mother cell or pollen mother cell, which undergoes meiotic division, okay, to form what is known as the microspore tetrad. Now, as and when, as I was explaining to you, as and when this anther starts to mature, okay, it starts to dehydrate, and these microspore tetrads, what happens is that they tend to dissociate from this tetrad condition, and later on they become what is known as, each one will become known as the pollen grain. Now, inside each of this microsporangium, you find millions and millions of pollen grains start to develop, and again simultaneously what happens is that these three layers, as I told you earlier, starts to dehiscence. That means they start to have a small opening because of dehydration, and these pollen grains, as we can see over here, see, there is no distinction over here right now. All these are pollen grains which are the result of this dissociation from the microspore tetrad, and they start to this is the dehiscence region. That means once when they were like this, now they have become like this. That means this is the place where the line of dehiscence is seen, and this line of dehiscence opens as the water dehydrates from the anther, okay, and these pollen grains starts to disperse out. Okay, so this is about the formation of pollen grains. Okay, next we talk about pollen grain itself.
Now let us talk about pollen grains. Take a flower, any flower for that matter, but highly accessible ones are such as hibiscus. And hibiscus has a visible anthers, and these anthers are colorful, especially yellowish. And when you touch them, you'll find a powdery kind of a substance sticking to your fingers. Now, these are pollen grains. Take a slide, dust these anthers again on them, and observe it under a microscope. You'll see beautiful, beautiful colored pollen grains as well. At about 25 to 50 micrometers in diameter, pollen grains are spherical in shape, and this pollen grain is made up of two layers. The outermost layer is called as the exine, and it is made up of the toughest substance known till date called as sporopollenin. Okay, and this exine all along contains small pores called as the germ pores. Now, this sporopollenin, that is what exine is made up of, is known to withstand the harshest acids and alkalis, and that's the reason they can be fossilized as well. Now, when you come to the innermost layer called as the intine, now this intine is made up of cellulose and pectin and is covered by plasma membrane. Now, at maturity, pollen grain is shed at two cell stage, as you can see over here. Right. Now, this is how it develops into two cell stage. Okay. And this is the two cell stage. Now, the two cells which are found in intine are that vegetative cell and the generative cell. Now, as you can see over here, vegetative cell is quite large and it has a dense cytoplasm and it has abundant food reserve. Whereas if you look at the generative cell, it is small, spindle shaped, and usually seen floating around inside the vegetative cell. Now, almost 60% of the angiosperms shed their pollen grains at this two cell stage, whereas the other species are known to shed the pollen grains when this generative cell divides mitotically to give rise to two male gametes. As much as these pollen grains are known to have a very vital role to play in the sexual reproduction of angiosperms, to mankind, for some people, they are known to be harsh. These same pollen grains are known to cause severe allergies and lung disorders, or, for example, if you can take asthma or some other allergies as well. Now, for example, if you can take Parthenium or carrot grass, which came to India with wheat, now that is known to cause severe allergies in few people. Then there are good things about this pollen grain as well. In Western countries, there is a new trend which is going on where we see that pollen grains are made up into capsules and syrups as well, and they are known to have lots of many vital nutrients in them, so they are consumed for good health. Now, if you take sports person and some race horses, even they are fed with or they are given this supplement and which are known to enhance their performance when they take it. Apart from this, pollen grains in some species they have what is known as the viability period. The viability period depends on species to species, where we see that some species, like for example rice and wheat, the pollen grains are known to be viable only for 30 minutes from the time they are shed from the stamens. Other varieties or other species such as Rosaceae is known to be the pollen grains of this are known to be viable months together from the time of shedding. So, as we see that we have what are known as the sperm banks, even we can start something known as the pollen banks, where we can store these pollen grains at minus 96 degrees Celsius or in a liquid nitrogen. So, these are the things about the male reproductive parts in a flower. Thank you.